by controlling different reacting conditions. And now let's start introducing our project. This is the outline. I will first introduce the main goal, motivation, and main idea of the E. coli tuner. And then we are going to talk about the mechanisms and functions with different regulators. There are light, temperature, and sRNA. After analyzing the regulators, we are going to show the diagram we designed and explain the mechanism of the regulating pathway. And then we'll move on the application part to introduce how the E. coli tuner should be used. Finally, we'll make summary of the E. coli tuner. And now let's start from our main goal. It is not a dream anymore that bacteria-engineered machine give us the opportunity to fulfill our mission. And so, we thought that one day, if we could have the ability to control the bacteria to work along with the transitions of environmental conditions, then we can give the bacteria a more accurate order. For example, we add different tags to the bacteria with special reacting condition designed. The only thing we have to do is to put them to the right place to do the right thing. We move to another condition. The former reaction stops and begins the new one. On the concepts, each works under a specific condition. Such regulating systems give us the opportunity to let the bacteria work accurately what we command, and we will only have to change the environment of reacting for the bacteria to work what we want. And now let's talk about our motivation. During daily life, changes of day and night, such natural and non-invasive regulators motivate us in designing the regulating systems. The light regulating system is different from the common regulating system. In common regulating system, we usually see in iGEM projects, we usually have to add extra substrates to induce the promoter. However, different from adding substrates to induce the promoter, the light promoter sends the specific wave-light directly to turn on the gene expression. Also, we don't have to concern about the concentration of extra substrates we add when turning on or off the promoter and the side effects on the bacteria. Besides the light, we also use two regulators, temperature and sRNA. We regulate the temperature when changing the different reacting conditions. By controlling these two non-invasive regulators, we can turn on or shut down the gene expression specifically. And we hope to achieve a regulating system that is flexible and adjustable. On the other hand, there are many applications referring to the sRNA acting mechanism. By comparing the base pair of sRNA with mRNA, we can turn off the gene expression specifically. So, our main idea is, by combining these three regulators, light, temperature, and sRNA, we can achieve a model that is cross-regulated and we can change the environment reacting for the bacteria. And by comparing these different specific conditions, we can order the bacteria to work more specifically along with how we want. We also use the sRNA inhibiting systems to turn off the unnecessary reactions. So, by using these non-invasive regulators, we hope that this regulating system could be used to any applications that require specific control. And we tend to make the bacteria-engineered machine a more accurate effect. So now, let's talk about our regulators.